Hey guys, welcome back to another episode of my channel. My name is Keika. I go over textbooks chapter by chapter, section by section. This video will be going over concept 6.4, the endomembrane system regulates protein traffic and performs metabolic functions. So let's get started. Many of the different membrane-bounded organelles of the eukaryotic cell are part of the endomembrane system, which includes the nuclear envelope, the endoplasmic reticulum, the Golgi apparatus, lysosomes, various kinds of vesicles and vacuoles, and the plasma membrane. The system carries out a variety of tasks in the cell, including synthesis of proteins, transport of proteins into membranes and organelles or out of the cell, metabolism and movement of lipids, and detoxification of poisons. The membranes of the system are related either through direct physical continuity or by transfer of membrane segments as tiny vesicles, which are sacs made out of membrane. Despite these relationships, the various membranes are not identical in structure and function. Moreover, the thickness, molecular composition, and types of chemical reactions carried out in a given membrane are not fixed, but may be modified several times during the membrane's life. Having already looked at the nuclear envelope, we will now focus on the endoplasmic reticulum and other endomembranes to which the endoplasmic reticulum gives rise. Okay, so for my notes here, I wrote the endomembrane system includes the nuclear envelope, endoplasmic reticular, or ER, Golgi apparatus, lysosomes, various vesicles and vacuoles, plasma membrane. This system carries out a variety of tasks, such as protein synthesis, transport of proteins into membrane or organelles or outside of the cell, metabolism and movement of lipids, and detoxification of poisons. This is done either through direct physical continuity or transfer of vesicles, which are sacs made out of membrane. Okay, so the endoplasmic reticulum, biosynthetic factory. The endoplasmic reticulum, or ER, is such an extensive network of membranes that it accounts for more than half the total membrane in many eukaryotic cells. The word endoplasmic means within the cytoplasm, and reticulum in Latin is Latin for little net. The ER consists of a network of membranous tubules and sacs called cisternae, from the Latin cisterna, a reservoir for a liquid. The ER membrane separates the internal compartment of the ER called the ER lumen, or cavity, or cisternal space, from the cytosol. And because the ER membrane is continuous with the nuclear envelope, the space between the two membranes of the envelope is continuous with the lumen of the ER. And here we have an image. We have an image of the nuclear envelope in the middle here, and you see it morphing into the endoplasmic reticulum here in blue. The little red dots here are ribosomes. See, ribosomes. Cisternae are the folds that you see, and the ER lumen it's the space in between the cisternae, or as they said before, the cisternal space, right? And we'll go over the transitional ER, transport vesicle, rough ER, smooth ER in just a second. There are two distinct, though connected, regions of the ER that differ in structure and function: smooth ER and rough ER. Smooth ER is so named because its outer surface lacks ribosomes. Rough ER is studded with ribosomes on the outer surface of the membrane and thus appears rough through the electron microscope. As already mentioned, ribosomes are also attached to the cytoplasmic side of the nuclear envelope's outer membrane, which is continuous with the rough ER. So for my notes, I wrote endoplasmic reticulum is a network of membranes that accounts for more than half the total membrane in many eukaryotic cells, consists of tubules and sacs called cisternae. ER lumen is a cavity in between the cisternae folds and is continuous with the nuclear envelope. ER membrane separates the internal compartment of the ER lumen or cisternal space from the cytosol. Smooth ER lacks ribosomes. Rough ER is studded with ribosomes on the outer surface of the membrane, appearing rough. Okay. Functions of the smooth ER. The smooth ER functions in diverse metabolic processes which vary with cell type. These processes include synthesis of lipids, metabolism of carbohydrates, detoxification of drugs and poisons, and storage of calcium ions. Enzymes of the smooth ER are important in the synthesis of lipids, including oils, steroids, and new membrane phospholipids. Among the steroids produced by the smooth ER in animal cells are the sex hormones of vertebrates, the various steroid hormones secreted by the adrenal glands, Among the steroids produced by the smooth ER in animal cells are the sex hormones of vertebrates and the various steroid hormones secreted by the adrenal glands. The cells that synthesize and secrete these hormones, in the testes and ovaries, for example, are rich in smooth ER, a structural function that fits the function of these cells. Other enzymes of the smooth ER help detoxify drugs and poisons, especially in liver cells. Detoxification usually involves adding hydroxyl groups to drug molecules, making them more water-soluble and easier to flush from the body. Kind of like diluting something to make it less dense and easier, like they said, easier to flush from the body. The sedative phenobarbital and other barbiturates are examples of drugs metabolized in this manner by smooth ER in liver cells. In fact, barbiturates, alcohol, and many other drugs induce the proliferation of smooth ER and its associated detoxification enzymes, thus increasing the rate of detoxification. This in turn increases tolerance to the drugs, meaning that the higher doses are required to achieve a particular effect, such as sedation. Also, because some of the detoxification enzymes have relatively broad action, the proliferation of smooth ER in response to one drug can increase the need for higher dosages of other drugs as well. Barbiturate use, for instance, can decrease the effectiveness of certain antibiotics and other useful drugs. The smooth ER also stores calcium ions, and in muscle cells, for example, the smooth ER membrane pumps calcium ions from the cytosol into the ER lumen. When a muscle cell is stimulated by a nerve impulse, calcium ions rush back across the ER membrane into the cytosol and trigger contraction of the muscle cell. In other cell types, release of calcium ions from the smooth ER triggers different responses, such as secretion of vesicles carrying newly synthesized proteins. Okay. So, function of the smooth ER. Functions. Synthesis of lipids, such as oils, steroids, new membrane phospholipids. Metabolism of carbohydrates. Detoxification of drugs and poisons. I marked drug tolerance. I think that's an important thing. I think they might ask a good question about drug tolerance, so just to make sure you understand that. Storage of calcium ions, muscles, and steroids, which are lipid hormones, produced by the smooth ER include sex hormones of vertebrates and steroid hormones by adrenal glands. Cells that synthesize and secrete these hormones are rich in smooth ER. Enzymes of the smooth ER from liver cells help detoxify drugs and poison. Okay, so detoxification involves adding hydroxyl groups to drug groups. Detoxification involves adding hydroxyl groups to drug groups, making them more water-soluble and easier to dispose from the body. Barbiturates, alcohol, and other drugs produce the proliferation or increase or reproduction of smooth ER and its associated detox enzymes, thus speeds the rate of detoxification. This increases tolerance to drugs. Higher doses are required to achieve particular sedative effects. So I made a little chart here. Now let's just say you had one beer. This is you prior to drinking anything, let's say. this is your first beer, and you had 2 enzymes that detoxify your alcohol in your body. The next time you drink beer, the same amount, you have more enzymes ready to detoxify your 1 beer. So you would probably need, so just given, I don't know the exact amount, but let's just say you need a little bit more. Like, let's say you need 2 beers in order to reach the same effect that you had with 1 beer before, just to make it less. So this is me explaining tolerance. So 1 beer increases smooth ER, which thereby increases tolerance and detoxification. Okay? So I hope you understand that. I would also double check with your professor to explain this a little bit better. This is just based off my understanding, and I'm a student, so this is just how I understood it based off the text. Since some of the detoxification enzymes have broad action, other drugs need higher dosages as well. Barbiturates can decrease the effectiveness of antibiotics and other useful drugs. So let's just say, like, your useful drug is an antibiotic or an antidepressant, right? You drink alcohol, and now there's other molecules competing for effectiveness in your body because you have a limited amount of receptors in your body that work with your drugs, the antibiotics or the antidepressants, as well as the alcohol, which is meant to produce sedative effects. So when you have 2 drugs competing, a useful one and a not so useful one, you find that your useful one may not have the best effect because there's not enough receptors to get all of them, right? So that's what they're trying to say with broad action, and what they're saying how a barbiturate use can decrease the effectiveness of other useful drugs. So if you're on other medication, be careful and be mindful that your medication may not work as well because you're taking other substances. Smooth ER also stores calcium ions. I think they'll want you to know that it stores calcium ions, but I don't know if they'll want you necessarily to know how it works with the muscles, because that's something that you'll need to know in anatomy and physiology more in depth. But this is a good start to understand that in the muscles, smooth ER pumps calcium ions from the cytosol into the ER lumen, and then when the muscle cells are stimulated by nerve impulses, motor nerve impulses, calcium ions go into the ER membrane, endoplasmic reticulum membrane, into the cytosol, and triggers a contraction of the muscle cell. In other cell types, release of calcium ions trigger different responses, such as the secretion of vesicles carrying newly synthesized proteins. Okay. Functions of the rough ER. Many cells secrete proteins that are produced by ribosomes attached to the rough ER. For instance, certain pancreatic cells synthesize the protein insulin in the ER and secrete this hormone into the bloodstream. As a polysaccharide chain grows from a bound ribosome, the chain is threaded into the ER lumen through a pore formed by a protein complex in the ER membrane. The new polypeptide folds into a functional shape as it enters the ER lumen. Most secretory proteins are glycoproteins, which are proteins with carbohydrates covalently bonded to them. and the carbohydrates are attached to the proteins in the ER lumen by enzymes built into the ER membrane. After secretory proteins are formed, the ER membrane keeps them separate from proteins in the cytosol, which are produced by free ribosomes. Secretory proteins depart from the ER wrapped in a membrane in the membranes of vesicles that bud like bubbles from specialized region called transitional ER. So we'll just go back to the figure. Oops. So here we have the transitional ER, and here we see a bubble starting to form from the membrane, and this is a transport vesicle. This is the bubble, that results from the ER. Vesicles in transit from one part of the cell to another are called transport vesicles. We will examine their fate shortly. In addition to making secretory proteins, rough ER is a membrane factory for the cell. It grows in place by adding membrane proteins and phospholipids to its own membrane. As polypeptides destined to be membrane proteins grow from the ribosomes, they are inserted into the ER membrane itself and anchored there by their hydrophobic portions. Like the smooth ER, the rough ER also makes membrane phospholipids. Enzymes built into the ER membrane assemble phospholipids from precursors in the cytosol. The ER membrane expands and portions of it are transferred in the form of transport vesicles to other components of the endomembrane system. Okay? So here for the functions of ER, rough ER. Many cells secrete proteins produced by ribosomes attached to the rough ER. Okay? So many cells secrete proteins produced by ribosomes attached to the rough ER. Pancreatic cells synthesize the protein insulin and secrete them into the bloodstream, for example. As polypeptide chains grow from the bound ribosomes, the chain is threaded into the ER lumen through a pore, and the new polypeptide folds into its functional shape as it enters the lumen. Many secretory proteins are glycoproteins, which are proteins with carbohydrates covalently bonded to them. Carbs are attached to the proteins in the ER lumen by enzymes built into the ER. After secretory proteins are formed, they leave the ER wrapped in membranes of vesicles that produce like bubbles from the specialized region called transitional ER. Transport vesicles are vesicles that transit from one part of the cell to another. Rough ER also produces the membrane for the cell by adding membrane proteins and phospholipids to its own membrane. Polypeptides fated to be membrane proteins from ribosomes are inserted into the ER membrane and anchored there by hydrophobic regions. Smooth and rough ER both make membrane phospholipids. Enzymes built into the ER membrane assemble phospholipids from the precursors, or parts, found in the cytosol, and the ER membrane expands, and parts are transferred in the form of transport vesicles to other parts of the endomembrane system. The Golgi apparatus, shipping and receiving center. After leaving the ER, many transport vesicles travel to the Golgi apparatus. We can think of the Golgi as a warehouse for receiving, sorting, shipping, and even some manufacturing. Here, products of the ER, such as proteins, are modified and stored and then sent to other destinations. Not surprisingly, the Golgi apparatus is especially extensive in cells specialized for secretion. The Golgi apparatus consists of a group of associated flattened membranous sacs, cisternae, looking like a sack of pita bread or pancakes. A cell may have many, even hundreds, of these stacks. The membrane of each cisterna in a stack separates its internal space from the cytosol. Vesicles concentrated in the vicinity of the Golgi apparatus are engaged in the transfer of material between parts of the Golgi and other structures. So, this is the Golgi apparatus. Here we see in number 1, vesicles move from the ER to the Golgi, so it's coming. And then here, the vesicles coalesce, or morph into, to form new cis Golgi cisternae. So we'll talk about cis in a second. And then 3, cisternal maturation. Golgi cisternae move in a cis to trans direction. So we'll talk about that in a second. But this is basically, I just wanted to give you guys an image of what it looks like. As you can see, it's like parallel folds in a way. They're right next to each other. A Golgi stack has a distinct structural directionality, with the membranes of cisternae on opposite sides of the stack differing in thickness and molecular composition. The two sides of the Golgi stack are referred to as the cis space and the trans space. These act respectively as the receiving and shipping departments of the Golgi apparatus. The term cis means on the same side, and the cis face is usually located near the ER. Transport vesicles move material from the ER to the Golgi apparatus. A vesicle that buds from the ER can add its membrane and its content of its lumen to the cis space by fusing with the Golgi on that side. The trans space, on the opposite side, gives rise to vesicles that pinch off and travel to other sites. Products of the endoplasmic reticulum are usually modified during their transit from the cis region to the trans region of the Golgi apparatus. For example, glycoproteins formed in the ER have their carbohydrates modified, first in the ER itself, and then as they pass through the Golgi. The Golgi removes some sugar monomers and substitute others, producing a variety of carbohydrates. Membrane phospholipids may also be altered in the Golgi. In addition to its finishing work, the Golgi apparatus also manufactures some macromolecules. Many polysaccharides secreted by cells are Golgi products. For example, pectin and certain other non-cellulose polysaccharides are made in the Golgi of plant cells and then incorporated along with cellulose into their cell walls. Like secretory proteins, non-protein Golgi products that will be secreted depart from the trans space of the Golgi inside transport vesicles that eventually fuse with the plasma membrane. The contents are released and the vesicle membrane is incorporated into the plasma membrane, adding to the surface area. The Golgi manufactures and refines its products in stages, with different cisternae containing unique teams of enzymes until recently. The Golgi manufactures and refines its products in stages, with different cisternae containing unique teams of enzymes. Until recently, biologists viewed the Golgi as a static structure, with products in various stages of processing transferred from one cisterna to the next by vesicles. While this may occur, research from several labs has given rise to a new model of the Golgi as a more dynamic structure. According to the cisternal maturation model, the cisterna of the Golgi actually progress forward from the cis to the trans space, carrying and modifying their cargo as they move. The reality probably lies somewhere in between the two models. Recent research suggests the central regions of the cisternae may remain in place while the outer ends are more dynamic. Sorry, my nose. Before a Golgi stack dispatches its products by budding vesicles from the trans space, it sorts these products and targets them for various parts of the cell. Molecular identification tags such as phosphate groups added to the Golgi products aid in sorting by acting like zip codes on mailing labels. Finally, transport vesicles budded from the Golgi may have external molecules on their membranes that recognize docking sites on the surface of specific organelles or on the plasma membrane, thus targeting vesicles appropriately. So for my notes here, the Golgi apparatus, shipping and receiving center. Transport vesicles leaving the ER enter the Golgi apparatus. Warehouse for receiving, sorting, shipping, and manufacturing. Products of the ER, which are proteins and enzymes, are modified, stored, and sent to other destinations. The Golgi is very extensive and cells specialized for secretion. Golgi apparatus consists of a group of flattened membranous sacs called cisternae. Looks like a stack of pita bread or pancakes. There are many, sometimes hundreds, of these stacks. Vesicles in the area of the Golgi apparatus transfer materials between parts of the Golgi and other structures. A Golgi stack has distinct structural directionality, with the membranes of cisternae of opposite sides of the stack differing in thickness and molecular composition. Cis space and trans space act as receiving and shipping centers. Cis, on the same side, located near the ER, receives transport vesicles from the ER to the Golgi apparatus. Trans on the opposite side gives rise to vesicles that pinch off and travel to other sites. Products are usually modified from the cis to trans region of the Golgi apparatus. Glycoproteins removes sugar monomers and substitutes others, and membrane phospholipids. Golgi apparatus also manufactures some macromolecules. Polysaccharides secreted by cells are Golgi products, pectins and non-cellular polysaccharides of plant cells. Non-protein Golgi products leave the trans space of the Golgi inside transport vesicles that eventually fuse with the plasma membrane, where the contents are released and the vesicle becomes part of the plasma membrane. Golgi apparatus manufactures and refines products in stages, with enzymes of different cisterna. Research has led to the cisternal maturation model, where the cisternae actually progress forward from the cis to trans phase, thus carrying and modifying the products as they move. Before shipping the products, the Golgi sorts and targets products for various parts of the cell via molecular identification tags, phosphate groups added to products, to aid sorting. Transport vesicles may also have external molecules on their membranes that recognize docking sites on the surface of their specific organelles or on the plasma membrane. Okay. Lysosomes. Digestive compartments. A lysosome is a membranous sac of hydrolytic enzymes that many eukaryotic cells use to digest or hydrolyze macromolecules. Lysosomal enzymes work best in an acidic environment found in lysosomes. If a lysosome breaks open or leaks its content, the release enzymes are not very active because the cytosol has a near-neutral pH. However, excessive leakage from a larger number of lysosomes can destroy a cell by self-digestion. Hydrolytic enzymes in lysosomal membrane are made by rough ER and then transferred to the Golgi apparatus for further processing. At least some lysosomes probably arise by budding from the trans space of the Golgi apparatus. How are the proteins in the inner surface of the lysosomal membrane and the digestive enzymes themselves spared from destruction? Apparently, the three-dimensional shapes of these proteins protect vulnerable bonds from enzymatic attack. Lysosomes carry out intracellular digestion in a variety of circumstances. Amoebas and many other unicellular protists eat by engulfing smaller organisms or food particles, a process called phagocytosis. From the Greek phagion, to eat, and ketos, vessel, referring here to the cell. The food vacuole, formed in this way, then fuses with a lysosome, whose enzymes digest the food. Digestion products, including simple sugars, amino acids, and other monomers, pass into the cytosol and become nutrients for the cell. Some human cells also carry out phagocytosis. Among them are macrophages, a type of white blood cell that helps defend the body by engulfing and destroying bacteria and other invaders. So, this is the food vacuole. Contains food inside. And then this is a lysosome that has digestive enzymes. Lysosome contains active hydrolytic enzymes, so these are the digestive enzymes. It morphs together. The lysosome fuses with the food vacuole, and then the hydrolytic enzymes that were inside the lysosome digest the food particles inside the vacuole, the new vacuole. Okay? Lysosomes also use their hydrolytic enzymes to recycle the cell's own organic material, a process called autophagy. Autophagy. A damaged organelle or small amounts of cytosol becomes surrounded by a double membrane of unknown origin, and a lysosome fuses with the outer membrane of this vesicle. The lysosomal enzymes dismantle the inner membrane and the enclosed material, and the resulting small organic compounds are released to the cytosol for reuse. With the help of lysosomes, the cell continually renews itself. A human liver cell, for example, recycles half of its macromolecules each week. The cells of people with inherited lysosomal storage diseases lack a functioning hydrolytic enzyme normally present in lysosomes. The lysosomes become engorged with indigestible material, which begins to interfere with other cellular activities. In Tay-Sachs disease, for example, a lipid digesting enzyme is missing or inactive, and the brain becomes impaired by the accumulation of lipids in the cell. Fortunately, lysosomal storage diseases are rare in general population. Okay, so for my notes, lysosomes, digestive compartments. Lysosomes are sacs of hydrolytic enzymes that many eukaryotic cells use to digest and hydrolyze macromolecules. Lysosomal enzymes work best in acidic environments, so if they somehow enter the cytosol, the enzymes won't be affected because cytosol is near a neutral pH. Excessive leakage from many lysosomes may destroy a cell by self-digestion. Hydrolytic enzymes in lysosomal membrane are made of rough ER and transferred to the Golgi apparatus for further processing. 3D shapes of these specific proteins are protected from vulnerable bonds from enzymatic attack. Lysosomes carry out intracellular digestion in a variety of circumstances, such as amoebas and other unicellular protists engulf small organisms or food particles through phagocytosis. Food vacuoles formed in this way fuses with a lysosome whose enzymes digest the food. Digestion products, such as simple sugars, amino acids, or other monomers, pass into the cytosol and become nutrients for the cell. Or human cells, such as macrophages, a white blood cell, also do phagocytosis by engulfing bacteria and other invaders, thus defending the body. Lysosomes also use their hydrolytic enzymes to recycle their own organic material. Autophagy? Phagy? I don't know. Autophagy. A damaged organelle or small amount of cytosol becomes surrounded by a double membrane, and we don't know how. And a lysosome fuses with the outer membrane of this vesicle. Enzymes dismantle the inner membrane and the enclosed material, and the result is that small organic compounds are released to the cytosol for reuse. Therefore, the cell can continually renew itself. People with inherited lysosomal diseases have cells that lack a functioning hydrolytic enzyme normally present in lysosomes. Tay-Sachs disease. The brain becomes impaired by accumulation of lipids in cells because lipid digesting enzyme is missing or inactive. Rare. Okay, now moving on to vacuoles. Vacuoles. Diverse maintenance compartments. Vacuoles are large vesicles derived from the endoplasmic reticulum and Golgi apparatus. Thus, vacuoles are an integral part of a cell's endomembrane system. Like all cellular membranes, the vacuolar membrane is selective in transporting solutes. As a result, the solution inside a vacuole differs in composition from the cytosol. Vacuoles perform a variety of functions in different kinds of cells. Food vacuoles formed by phagocytosis, food vacuoles formed by phagocytosis have already been mentioned. Many unicellular protists living in fresh water have contractile vacuoles that pump excess water out of the cell, thereby maintaining a suitable concentration of ions and molecules inside the cell. In plants and fungi, certain vacuoles carry out enzymatic hydrolysis, a function shared by lysosomes in animal cells. In fact, some biologists consider these hydrolytic vacuoles to be a type of lysosome. In plants, small vacuoles can hold reserves of important organic compounds, such as the proteins stockpiled in the storage cells and seeds. Vacuoles may help protect the plant against herbivores by storing compounds that are poisonous or unpalatable to animals. Some plant vacuoles contain pigments such as red or blue pigments of petals that attract pollinating insects to flowers. Mature plant cells generally contain a large central vacuole, which develops by coalescence of smaller vacuoles. The solution inside the central vacuole, called cell sap, is the plant cell's main repository of inorganic ions, including potassium and chloride. The central vacuole plays a major role in the growth of plants, which enlarge as the vacuole absorbs water, enabling the cell to become larger with the minimal investment in new cytoplasm. The cytosol often occupies only a thin layer between the central vacuole and the plasma membrane, so that the ratio of plasma membrane surface to cytosolic volume is sufficient, even for a large plant cell. Okay, so for vacuoles, I wrote, vacuoles are large vesicles derived in the ER and Golgi apparatus. The vacuolar membrane is selective in transporting solutes, and as a result, the solution inside the vacuole is different from the cytosol. Variety of functions. Food vacuoles formed by phagocytosis, contractile vacuoles that pump excess water out the cell to maintain concentration of ions and molecules in the cell. Plants and fungi have vacuoles that do enzymatic hydrolysis, and some biologists think hydrolytic vacuoles are a type of lysosome. In plants, small vacuoles may hold reserves like proteins and storage cells. And vacuoles can also protect a plant by storing compounds that are poisonous or unpalatable to animals. Vacuoles in plants may also have pigments attracting pollinating insects to flowers. Mature plant cells generally have a large central vacuole by coalescence joining of smaller vacuoles. The solution inside, called cell sap, is the plant cell's main repository of inorganic ions, including potassium and chloride. Major role in growth of plants. Enlarges as the vacuole absorbs water. Animal cells don't have this. So, the endomembrane system, a review. Figure 6.5 reviews the endomembrane system, showing the flow of membrane lipids and proteins through the various organelles. Flow of membrane lipids and proteins through the various organelles. Yes. As the membrane moves from the ER to the Golgi and then elsewhere, its molecular composition and metabolic functions are modified, along with those of its contents. The endomembrane system is a complex and dynamic player in the cell's compartmental organization. So here we see this chart. I think this is a really helpful chart. The nuclear envelope is connected to the rough ER, which is continuous with the smooth ER. So here we see the nucleus and the nuclear envelope. The smooth ER, which is on the outside, the rough ER on another side. And then here we see membranes and proteins produced by the ER move through transport vesicles to the Golgi. So this is a transport vesicle. And here it merges, coalesces, as they say in this textbook, into the Golgi apparatus from the cis phase first. And then the Golgi pinches off transport vesicles and other vesicles that give rise to lysosomes, other types of specialized vesicles and vacuoles. Okay, so here from the trans phase, it's pinching off, and we have a lysosome, or yeah, I guess. Well, this is just a review. Like, it can either send off proteins or other material, or it can be a lysosome, which the lysosome is available for fusion with other vesicles for digestion. Or it's a transport vesicle that carries protein to the plasma membrane for secretion, such as this one. And number 6, right over here, the plasma membrane expands by fusion of vesicles, and proteins are secreted from the cell by exocytosis, which is exocytosis. Let's just look that up real quick. Exocytosis. Cellular process of transporting molecules out of the cell, right? Out of the cell. We'll continue with our tour of the cell with some organelles that are not closely related to the endomembrane system, but play crucial roles in the energy transformation carried out by cells. So, here I made my notes, the endomembrane system a review. And I basically wrote everything that was within this chart. The nuclear envelope is connected to the rough ER, which then connects with the smooth ER. Membranes and proteins produced by the ER move through the Golgi apparatus via transport vesicles. The Golgi pinches off transport vesicles and other vesicles that develop into lysosomes, other specialized vesicles, and vacuoles. The lysosome fuses with other vesicles for digestion. Transport vesicles carry proteins to plasma membrane from secretion, and plasma membrane expands with the fusion of vesicles, while proteins are secreted out of the cell, exocytosis. Okay, so next video we'll be going over concept 6.5, mitochondria and chloroplasts change energy from one form to another. Hope you guys understood the material better as a result of this video. Sorry it's been a really long time. I have excuses, but I don't think you want to hear them, so it's okay. I'll see you guys next time, and I hope you guys have a wonderful semester. Take care. Bye-bye.